need to come up with new methods to control invasive species because basically the current methods have side effects. They tend to often harm unintended species that we want to keep safe. And so the hope with these new genetic methods of biocontrol is that we can be more precise.
What I consider genetic biocontrol is any technology that essentially uses genetic engineering to convert the pest organism into the pesticide. Genetic biocontrol technology in carp works by engineering carp to spread deleterious genes through the carp population, and that happens through mating.
Ideally, we wouldn't have to treat the lake a million times. Right? We just have to release the control agent into the lake, and it would become an established part of that ecosystem and create the control that we need for a long, sustainable amount of time. Genetic biocontrol comes in two flavors. There's the DNA-based version and the RNA-based version. The DNA-based version, I would say, is preferable. It's further along in terms of the science. The only disadvantage to it is that it requires that we be able to breed the animals in captivity.
So one of the challenges that we have in applying these tools in zebra mussels, despite the fact that they grow and reproduce extremely well in the wild, in the lakes and rivers, ironically, we're not able to cultivate them over the long term in the lab. And so we're trying to use an approach called RNA interference, where we can genetically manipulate microbes to express double-stranded RNAs and then feed those to zebra mussels to introduce the genetic biocontrol tool without actually manipulating the zebra mussel itself.
Studies have found that most people have either a little bit or no knowledge at all about genetic biocontrol. And so it's important to keep this in mind when we go forward and we start engaging the public on genetic biocontrol. So we may need to educate them on what genetic biocontrol is.
You don't want to spend years developing a genetic biocontrol fish that is never going to get used because the public would never accept it the way that you designed it. So making sure that it's safe and meets, the requirements of what the public is okay with is very important early on.
The order of operations as we see it now for bringing genetic biocontrol technology to the field is you start by making a proof of concept in the lab, you study it in the lab, then you'll bring it outside to a controlled, contained pond trial where you introduce it to things like environmental fluctuations, and from there you go to a small-scale field trial, and then a larger-scale field trial before it's ready for prime time.